Hello to our friends joining us via recording. Today we are starting our first lesson in unit number 5, and that is lesson number 5. Before we move in to really discuss lesson number 5 though, we wanted to take a moment because we just finished exam number 1. We're done with unit number 1. We wanted to compare notes, if you will, together about the things we did to prepare for our exam that were the most helpful and the least helpful. So I'm going to do the typing for us here. I made us a nice little chart. If you can help me out, my friends that are here in class, in the chat, can you tell me what were some of the things that you did that were the most helpful for you? As you were preparing for exam number 1, what helped you prepare for that the best? Okay, so one of the notes that I'm seeing is those practice assignments. The practice assignments, yeah, somebody else is typing too. Getting in those practice assignments or the practice exams, absolutely. I know somebody is mentioning the review session. I'm glad to hear that was helpful for us. And yeah, in conjunction with that, taking notes during class, absolutely. I think one of the best things to do, hopefully we've kind of figured this out with our class too, is filling out your outline before class and then bringing it to class already completed, so you're not looking for blanks or any of that stuff, and you can just add notes to it. That's what I think might be the most effective way to do our class together. So taking notes from class and using those guided lessons ahead of time so that these classes are most helpful for us. So yeah, I'm seeing a lot about the practice exams, doing those review assignments daily. That's perfect. I see some notes too about flashcards. Awesome. Yeah, rewriting the notes. So here, let me put a little information here about the notes. So we said complete before class and rewriting them. And my caveat that I'd say with the rewriting the notes, what I think is most helpful when you're rewriting them is putting it in your own words. I think that's really helpful too, finding new ways to say the same things. Because as we saw on the exam, Dr. Ellis gets sneaky, right, on the exams. So it's not going to be exactly the same way it was in your notes. It's not going to be exactly the same way that it was when you did the assignment or when we talked about it in the review. So yeah, the more you can relate it to real life stuff, the more that you can put it in your own words. All of those things are definitely helpful for sure to prepare for the exam. Let me check and see. Yeah, repeating things. I like that. Definitely. And yeah, just going through and reviewing, relating it to real life. Absolutely. Okay, so we've got some good ideas over here of how we prepared well for the exam. Now let's transition over to some of the things that we did that weren't so helpful for us. What are some of the things, now that you've taken the first exam, what are some of the things that you've done in the past to prepare that maybe you're not going to do anymore? Or what are you planning to change as we go into unit number 2? What were some of those least helpful things? Any ideas? Yeah, so Kaylin said her studying wasn't as helpful as she wanted it to be. We'll find new ways, Caitlin. We'll make studying more helpful. Yeah, I am glad to see that Summer mentioned one of the big ones, the one that comes up pretty much every semester. The thing that is going to help you the least is procrastination. Do not put it off. Don't wait until the last minute to start doing those homework assignments, to start learning the material. It has to kind of be a daily thing, like some of you said. Yeah, Fanshin's mentioning skipping over those small details. So some of those little important details, not focusing on those, can come back to bite you on the test, right? Hey, I'm pretty sure that we all knew the salty banana details though, right? Like if you've ever come to class, you knew the small details of salty banana. So that's one small detail we didn't forget, right? I like the comment that Anisha made about flashcards. Yeah, because flashcards kind of here. I'm gonna give them a little asterisk over here because she's totally right. She said that she could never finish them to actually study them. So that's the challenge with flashcards. It kind of ties into procrastination. Flashcards can be super helpful if you make them far enough in advance that you actually get to use them. Because if you wait until it's too late to make them, you'll have this whole awesome pile of flashcards and you never even get to look at them once. I think back to when I was in grad school, and I can't remember, I think it was like a biochemistry class or something ridiculous that I was taking. And I decided the night before the exam that the best thing for me to do was to make note cards. But I literally didn't get through all the content making note cards for it. And I literally didn't look at all the note cards I made. So if you're a flashcards person, if writing them out helps you, please make sure you're doing it as we go. Because doing it the night before, or even just a couple days before, you're not going to be able to use them in the way that you want to. So give yourself plenty of time. Flashcards can be a great tool, but they can also be a great way for us to waste time right before the exam when we should be studying. So we got to balance it. Let's see what else we got here. Yeah, I 100% agree with snowpocalypse. I agree with that one 100%. The good news is, knock on wood, I got some wood here. Knock on wood, we shouldn't have that one coming up for the lab midterm or any of our other exams. So fingers crossed, no more snowpocalypse. That would be great. I think we can all agree. Let's see. Looking at Sarah's comments, she mentioned that sometimes she only watched or listened to the lectures and didn't really pair that up with the notes outline. That wasn't helpful. So I'm going to call that, let's see, I'll call that passive learning. I'll put learning in quotes here. As much as I love to talk, and I know that it is just so wonderful to you all to listen to me for an hour and a half or 2 hours straight, it's not the best way to learn. So make sure we're taking notes as we go. I think that will help a lot too. Yeah, there is, so Audrey was mentioning the volume of information. Yeah, so finding ways to chunk up the information, because there's pretty much always going to be about 4 lessons on the exams. When we get to the units that have fewer lessons on the exams, they're actually going to be longer lessons. So it's always going to be about the same amount of content, unfortunately. So finding ways to avoid the procrastination. Obviously, we're in a good place because we're not going to get more snow, so we can avoid that one. And we've got some ideas of how to do more of the active learning to help us stay out of that track. So hopefully these ideas are helpful for us going forward. Let's try to lean on some of these things that we did that were helpful. The other thing I'll add, I mentioned it, I think before the recording. At the top of each of your notes outlines, there's a list of things called the learning objectives. These are the things that once you've studied the material and you've learned it and you understand it, these are the things you should be able to do. So I want to remind you, as maybe one other study tip to try for unit number 2, once you've gone through and you've taken your notes, and once you've done some of those practice assignments or you've gone to the review session, check out those learning objectives too and see if you can mentally organize your information from each of those classes into these learning objectives. So for today, we're doing lesson number 5, and you can see on the first page of that notes packet at the very top, the things I expect you to do. So you've got those words that you want to make sure you know what they mean. You've got lists of things where I tell you to identify things or describe things. If you can do that with your information, you are in good shape for the exams. Because here's a wink, wink, nudge, nudge. When I write the exams, I literally pull up those learning objectives in one window, and I pull up Blackboard in the other window, and I write my questions based on what those learning objectives say. So if you've ignored them up to this point, and they're like, man, that's just a waste of space, Here's a wink, wink, nudge, nudge. Check those guys out at the top of every lesson. If you're someone that likes to rewrite the notes, maybe rewrite them based on those learning objectives. So make yourself a new document where you organize that information around the learning objectives. So we're moving on from exam number 1. We're moving into unit number 2. Shoot me an emoji. What are our thoughts about moving into unit number 2? How are we feeling? What are we thinking? I got some thumbs up. Hey, that's great. Yeah, happy to have made it this far, right? Yeah, let's do it. Audrey says, let's bond some more, as long as it's not chemical bonds, right, Audrey? We want to move on from that chemistry. We're leaving that behind us. Alas, I cannot make there be fewer tests, but I will do my best to give us more penguins. I'll try to make us have more penguins. All right, well, let's dive into lesson number 5 then. A question in the chat about whether exam number 1 is one of the easiest exams. That's really hard to say. I think in terms of the volume of content, it might be one that has less volume of content on it, but it's your first time learning to take tests with me. So you are learning how I make you use information. I would suspect by the time we get to exam number 3 and number 4, when you come back and review exam number 1 with me, you'll be like, oh, that was so easy. I suspect that's how you'll feel by the time we get to exams like 3 and 4, when we're in those later units. That doesn't mean I think you're there right now, because you're getting used to it. So I would say I would say they're probably all maybe about the same. So in different ways. Audrey asked about the average. I honestly haven't checked. I think it was in the high 70s was the average, which is pretty standard for a first exam in my class. So and if, by the way, I think I'd mentioned this, but if exam number 1 didn't go well for you, that final exam, if your score is higher on the final exam than it was on exam number 1, I will actually replace your final exam score, use that score to replace exam number 1. So keep in mind that if this was a bad test for you, or it was secretly an ugly test for you, we can replace this score at the end of the semester. So all hope is not lost. All right. Unit number 2. We are going to spend a bunch of time here at the beginning talking about proteins. So today we're going to talk about the anatomy of proteins. Tomorrow and into next week, we're going to talk about how your cells build those proteins, the actual process of building them. As we finish up proteins, then we're going to transition into our first organ system, which we've already done in lab, and that's the skin. So this unit is a little bit, we bounce around a little bit as we start doing organ systems, but here at the beginning, we're really going to talk small picture. We're going to talk about proteins. When we talk about proteins, one of the first things that you should always think of anytime you hear the word protein is you should be thinking amino acids. Amino acids are what we call the monomers of proteins. The monomers of proteins. A monomer is a fancy word basically for saying the pieces. If you think about, one of my students told me they like to think about amino acids kind of like those little Lego pieces. So you've got these little Lego pieces, these amino acids, that we bring together to build our big thing, which is a protein. So we would call proteins a polymer. Polymer. Poly means a lot. Mono means one. So the monomer, the small piece that builds this really big polymer, which is a lot of pieces of a protein, that monomer is called amino acids. Now, this is my failed attempt at humor over here. Amino acids are oh so mean, right? They're actually like several of you were thinking when you took the exam, right? This is you staring at your computer screen saying, I don't like you, being an amino acid. The name of amino acids, though, it actually comes from the two parts of it. So this amino part of its name comes from the fact that over here on the left, we can see what's called an amine group. Now, for the record, I will never make you draw this structure. But we do need to know what these parts are. So my part over here with the nitrogen is called an amine group. My part over here on the right side with the carbon is the acid group. Technically, the type of acid this is, it's called carboxylic acid, carboxylic acid. So over here, I've got a carboxylic acid group. Over here, I have an amine group, and this is the structure of an amino acid. When I want to make a protein, when I want to make a polymer that has more than one amino acid in it, I'm actually going to interact the amine group of one of those amino acids with the acid group of the other. And so I'll show you how we do that in just a minute. But here's something that I want to point out. Notice how I've got a nitrogen here that's interacting with some hydrogens over here, a nitrogen and some hydrogens. We talked about how when water interacts with hydrogen, when we have water, so H2O, did oxygen share the electrons equally or unequally when it was oxygen and hydrogen in water? Does anyone remember? Did we share equally or unequally? Yeah, we shared unequally. Remember that water has dipoles, has those partial charges. So just like water, when I have an amine group over here, a nitrogen with a couple of hydrogens, this is also an example of a polar covalent bond. So what's going on between the hydrogen and the nitrogen, this is also polar covalent. So this is a part of my amino acid where we're not sharing electrons equally. And we kind of got the same thing going on over here as well with my oxygen and my hydrogen. So we're not sharing those electrons equally. We've got some polar covalent bonds or some dipoles going on. All right, so my goal is to build proteins. Our class today is about proteins. It's not just about amino acids. It's about the big polymer that is a protein. When I want to build a protein, a polymer with lots of things in it, I'm going to use a process called dehydration synthesis. Dehydration synthesis. This, by the way, is an underline highlight star word. This is an important idea for us to understand. To understand it, we're going to break it into two parts. Let's start at the end with this word synthesis. When you hear the word synthesis, what are some of the things that you think about? If I'm doing synthesis, what am I doing? It's kind of a mean question, right? If I'm doing synthesis. Yeah, I like it. I'm creating something, I'm making something new, right? So I'm going to simplify it to say that we're building something. I like the idea though of creating or generating something new. Absolutely. To build something, and in the case of our lecture today, to build a protein, we use the process of dehydration. Hey, we should all be familiar with dehydration because this is something that we talked a little bit about last unit, right? When we were talking about water intoxication, or the opposite is what's going on here with dehydration. When you're dehydrated, what's going on with the amount of water in your body? if you're dehydrated, what's happening? Yeah, exactly. Dehydration, you don't have enough water, right? So dehydration, water leaves essentially. When you're dehydrated, you're losing water. You're sweating it out, you're releasing it in urine, dehydrated. When I want to build a protein, I want to do this protein synthesis stuff here. When I want to build a protein, the way I'm going to do that is I'm actually going to dehydrate my amino acids, or I'm literally going to take away water. Now, it's not like the water that's floating around in the environment in our cells. When I'm building a protein, the water I'm going to take away is one amino acid's OH group right here. So this acid group that we just talked about before, I'm going to steal its OH. I'm going to steal, it's called a hydroxyl. Take this away from one amino acid. I want to connect it to another amino acid. So what I'm going to do is take away this hydrogen from the amine group on my amino acid. So I'm dehydrating, or I'm ultimately taking away 2 hydrogens and 1 oxygen. So I'm pulling off water. And by doing that, I end up connecting this carbon to this nitrogen. The process of dehydration synthesis. I steal water from these amino acids to build a bond. Dehydration synthesis. Dehydration synthesis. This is on our list of words that when we hear it, we're thinking proteins. Another word that's on our list of when I hear this, I'm thinking proteins, is amino acids. Amino acids are what I'm putting together using dehydration synthesis. One other really important term though, and this is again an underline highlight star, something we got to know. This right here, peptide bonds. Underline highlight star. The only kind of molecule in your body that has the special kind of bonds called a peptide bond is a protein. Peptide bonds are what I actually build when I do dehydration synthesis. When I put together 2 amino acids, I build a peptide bond. That is how I build proteins. So proteins, my polymer, are made of lots of monomers, small pieces, that are called amino acids. Those amino acids are held together by what we call peptide bonds. Peptide bonds made in dehydration synthesis to help us form a line of amino acids to help us start building our protein. What we just discussed is what's called the primary structure. The primary structure of a protein. Now, let me back up a moment and ask a question here. We're talking about structure, or what's there in a protein. When I talk about the structure of something, is that an example of anatomy or physiology? When we talk about what's there, structure. Yeah, exactly. We're talking about the anatomy of our proteins here. When we talk about protein anatomy, there's actually 4 different levels of structure, or 4 different levels of complexity. So every protein at its most basic level always starts with primary structure. We always start with this string of pearls, like Kayla mentioned, or this line of amino acids. Every protein has primary structure. Primary structure, if you were to describe it in the easiest possible words, it's basically just the order that our amino acids are attached to each other. So we've got, notice on my picture here, here's an amine group, and here's an acid group. This is one amino acid, and I used a peptide bond to connect it to another amino acid that I see right here, and I used another peptide bond to connect that to another amino acid, the order that my amino acids are in, the way that I did dehydration synthesis and built all my little peptide bonds, that forms the primary structure of my protein. Here's an important idea to make sure that we write down. The primary structure of a protein has to be right for all the other levels of structure to be right. The primary structure of a protein has to be right for all the other levels of structure to be right. To put it another way, if I mess up the order that these amino acids are in, I'm not going to be able to fold them correctly. And if I can't fold them correctly, I don't have the structures we're getting ready to talk about. So arguably one of the most important levels of structure in a protein is its primary structure. We've got to have the amino acids in the right order for us to be able to fold them correctly. The first way those amino acids fold are into what we call an alpha helix or a beta sheet, a beta pleated sheet, as it's called here. The alpha helix and the beta pleated sheet. Think about an alpha helix kind of like those ribbons you sometimes put on a gift. So it's called curling ribbon, where you can buy it at the store and it's straight, and then you rub it along a pair of scissors and it looks all curly, and you can pull it up and down and it bounces. That's one of my levels of what we call secondary structure, my first level of folding. The other secondary structure that we can have, though, is a beta pleated sheet, and that's a little bit more like an accordion. So where parts of this, as you can see here, kind of fold up and down on each other, that's what a beta pleated sheet looks like. Now, this, like I mentioned, is what we call the secondary structure of a protein, the secondary structure. Secondary structure is a kind of folding that's really localized, or it's little tiny parts of your amino acid chain that fold together. So I'm going to add to our list here the word localized. It's just small sections of your protein chain that fold this way. When we talk about the secondary structure of a protein, it's important for us to realize which parts of our amino acid are involved in forming this structure. I'm going to do a bad job here, but I'm going to try to draw us an amino acid. Okay, so over here is my acid group. I'm just going to write the letters. Here's my acid group. And over here is my amine group. And there's another little thing we're getting ready to talk about called my R group. That's down below. When we talk about the secondary structure of a protein, the parts of the protein that are making us fold this way are the amine groups interacting with the acid groups. So we can see that if we look over here, it's most clear over here. See how I've got an N and an H, an amine group, that's interacting with my acid group in a nearby amino acid. These two amino acids aren't neighbors. They're not right next to each other in the line, but they're close enough that the positive dipole over here, because nitrogen doesn't share, it's not playing nice here, we've got a positive dipole on one amine group that's attracted to the negative dipole in this acid group over here. I have formed what kind of bond? What kind of bond did I make between this positive dipole and this negative dipole? Do we know? Yeah, Kaylin actually guessed correctly for us here. We've got a hydrogen that has a positive dipole and an oxygen with a negative dipole. This is a hydrogen bond, a hydrogen bond. So when we think about secondary structure, the kind of bond that we formed because of hydrogen bonds. Hydrogen bonds. A positive dipole interacting with a negative dipole. And look, I've got another interaction right here, another hydrogen bond. Positive dipole and negative dipole. They're interacting with each other. And then I see some more down here, some more hydrogen bonds and some more hydrogen bonds. The same thing is happening over here with my alpha helix. It's still a positive dipole interacting with a negative dipole. But the way that this amino acid chain curled on each other is a slightly different shape than the way that this one curled on each other. It all comes back to our favorite chemistry, right? Or our least favorite chemistry, we're more honest. The positive dipole with the amine group, the negative dipole here in the acid group, when they get attracted to each other, depending on how far apart the amino acids are, they're going to form hydrogen bonds that will either make part of my chain fold like an accordion or make it fold up like these curlicue ribbons over here. So secondary structure, this is all about those amine groups interacting with the acid groups. They form hydrogen bonds with one another to fold us. By the way, let's remind ourselves, primary structure, my order of those amino acids, it's not hydrogen bonds that hold those amino acids together. What kind of bond was it that we said held together amino acids? What's that special kind of bond that makes my beads on a string? Exactly. Yep, it's those peptide bonds. So primary structure, make a note since we're making lists here. Primary structure, we said, was the order of the amino acids, and that one forms because of peptide bonds. Peptide bonds. Okay, so every protein starts by putting those amino acids in the correct order, putting them in a line. We connect them with peptide bonds. If the dipoles in some of those amine groups and acid groups get attracted to each other and form hydrogen bonds, we might make secondary structure. First we have primary, that's my beads on a string. Then we have secondary, that's an alpha helix or a beta sheet. Those alpha helices and beta sheets fold up on each other to make a protein that looks like this. So this is the appearance of a typical protein. So this is a protein that's actually involved in helping a cell know when it's time to die. It's got this particular appearance here. Before I move on and talk about what's called the tertiary structure, I want to pause for a moment. When I look at this protein, notice how I see my curlicue right here. I got a little curlicue up here. That's an example of what kind of structure? These curlicues that we're seeing here. Do we remember what those were called? Those fancy ribbons? Yeah, those are the alpha helices, right? The alpha helix, absolutely. Alpha helix, which is an example of a secondary structure. Secondary structure. And then I've got my accordions. They don't look quite as much like an accordion here, right? But I've got those things called the beta sheets. The beta sheets, those are also an example of secondary structure. Once a protein is done having its amine groups and its acid groups interact with each other, It'll take all of those secondary structures and the places where there aren't secondary structure, that's these gray lines here, takes all of that and kind of twists it together into a 3D shape. So if we were in class together, I would act it out for you. So imagine in front of you, you've got a line of beads. That's the primary structure. And then if you took those beads and kind of curled them up on each other back and forth on your table, and then you took all of that and squeezed it together kind of in the shape of a can. That's what we got going on with tertiary structure. This is the overall 3D shape of a protein. So the way that the protein folds when we put together those alpha helices and when we put together those beta sheets, all of that combined makes the tertiary structure of a protein. Every single protein has tertiary structure. We must have a 3D shape to do a job. So everybody has at least tertiary structure. Some of the important proteins in the body that we'll talk about in a bit actually do more than just tertiary structure. Everybody, though, at least has a 3D shape. We all got to have some kind of shape. Let's talk about how we decide what that 3D shape is going to be. So now we're going to talk about how we got from, back up, these alpha helices and these beta sheets, how we went from them to this whole thing here. And the way that we build our tertiary structure is because of the part of the amino acid that we didn't talk about yet. The last part in amino acid right here. We talked about the amine group. We talked about the acid group, how these are what I use to form my peptide bonds to connect things together. We talked about how the amine group and the acid group are what interact with each other to form the secondary structures, the alpha helices and the beta sheets. This functional group, like a couple of us have said in the chat, let me label it. The functional group is the part of the amino acid, this little R right here, is the part of the amino acid that's going to determine how I fold into a 3D shape. The reason that this functional group gets to decide how we fold into a 3D shape is because of all that stupid chemistry we just can't leave behind. When we talk about amino acids in proteins, there are kind of three main types of amino acids. Some of my amino acids have a functional group, or on this table they're calling them side chains. Same thing. Some of my functional groups are non-polar. Some of my functional groups are polar. Some of my functional groups are charged. They have a negative or they have a positive. Three types of functional groups that amino acids can have: polar, non-polar, or charged. Okay, let's go back. Before we talk about what it means that these amino acids are non-polar, please make sure you underline, highlight, star the note that you should never, ever, ever memorize which kind of side chain or which kind of functional group these amino acids have. I am never going to ask you on a homework assignment or on an exam if valine is polar, non-polar, positive, or negative. I'm never going to ask that. I will ask you to use information about the fact that valine is non-polar to make some predictions, but please do not memorize what these chains look like, and please do not memorize what group they go into. I will kind of information. But we do need to know some things here about the fact that they're non-polar. So let's talk about what we do need to know with these being non-polar. What are some of the things, when you hear that word nonpolar, what are some of the things that you think of when we hear nonpolar? Perfect. Okay, so some of us are hopping on that synonym. Synonym for nonpolar is hydrophobic. Perfect. Yep, so we've got no charge. Hydrophobic, as someone mentioned for me, means we are water fearing. Water fearing. Absolutely. Yep. And with the no charge thing, it's because we're sharing equally. Absolutely. Okay, so check this out. I tell you that a protein has an amino acid that is non-polar inside of it, a non-polar amino acid. You know that that amino acid shares its electrons equally. There's no charges going on with it. Most importantly, though, what you know with this being a non-polar amino acid is that it's afraid of water. Yeah, it would do great in the plasma membrane. Plasma membrane is hydrophobic in the middle, right? This amino acid, though, hates water. So when I start thinking about how I'm going to fold this protein into the correct shape, these amino acids, all of these ones that are nonpolar, these ones are like, yeah, don't put me next to water. I can't even with water. Don't put me there. So nonpolar amino acids will always be found in the inside of a protein where it wouldn't be exposed to water. Nonpolar amino acids are hydrophobic. They can't even with water. We have other amino acids that are polar. What are some of the things you can tell me about polar amino acids? Let's make our list with polar amino acids. What's some of the stuff that name tells us? Yeah, so polar means that we are unequal sharing, absolutely unequal sharing. So we're going to have those dipoles. And things that have dipoles are hydrophilic. They are water-loving, absolutely water-loving. All of these amino acids that are listed as polar amino acids, all of these are hydrophilic. All of them have dipoles, those partial charges. When I'm trying to fold a protein into a 3D shape, my polar amino acids say, oh yeah, put me by water. I love water. So on the outside of a protein, we're going to find a lot of polar amino acids because polar things play nice with water. They like to be next to water because just like water, they've got dipoles too. So non-polar amino acids typically going to hide in the middle of a protein where there's no water. Polar amino acids more likely to be on the outside of a protein that's right next to water. When we talk about our negatively charged or positively charged amino acids, these ones also have no problems with water. Remember, it's like when we talked about with salt, how salt has an ionic bond. In an ionic bond, the positive of sodium is attracted to the negative of chloride, and that's what holds them together. The opposites attract. But when you put salt in water, it falls apart. It loves to be around water though. So same thing with these positive and negatively charged amino acids. They don't mind if they're by water because water has a dipole that keeps them happy too. These functional groups come in a whole bunch of different varieties. Polar, nonpolar, positive, negative. This is the part of an amino acid that's most responsible for the 3D shape of a protein, the way that it folds all the way. That's the job of these functional groups, these R groups. Here's the many different ways that these functional groups can interact with each other. So let's start here at the top. Before I go on, I need to ask a question here in the class. For any of my friends who are here in class today, do I have any Tom Brady fans? Because I'm about to offend you if you're here today and you like Tom Brady. Any takers? Okay, Chris doesn't even know who that is. That's good. Okay, so Tom Brady is a football player. Yeah, so he's a football player. He's actually the quarterback that just won the recent Super Bowl. He was with the Patriots forever. So I would always crack this joke, especially last year was the last time I got to crack it in person, alas, about how there's two kinds of football people in the world. There's the people who root for Tom Brady, and there's everybody else. So when I was joking with students when I taught this lesson last spring, it was like, so it doesn't matter who's in the Super Bowl. Whoever is playing against Tom Brady is the person I'm rooting for. Like, it's just the way it is. He's not my favorite. But what was really funny is the last time I did this in class, I actually had a student, he raised his hand when I asked if there were any Patriots fans, because back then Brady was with the Patriots. And I said that I apologized in advance. And then I still cracked the same joke. The next like 3 weeks when I met with that student, he was always wearing his Brady jersey. It was really funny. Here's how that absurd analogy with a Tom Brady jersey works out though. Some parts of your protein are going to fold together because all of those amino acids that are next to each other in this area, all of them are hydrophobic. All of them are hydrophobic. What does this word hydrophobic mean again? Hydrophobic. Yeah, we're afraid of water. We don't like water, right? We don't play nice with water. Hey, just like me and many football fans in the United States, we might all root for different football teams normally in the regular season. But when it comes time for Super Bowl season, we're all rooting against Tom Brady. I'm a Broncos fan, just gonna be real here. I'm a Broncos fan. I root for the Broncos. My sister-in-law is a Chiefs fan. That's a little point of contention. Not actually, but we like to joke. So she's a Chiefs fan. During the regular season, she votes for the Chiefs. I root for the Broncos. When it comes time for the Super Bowl, though, I don't care who's there, and she doesn't care who's there as long as it's not Tom Brady. So this idea of hydrophobic attractions. We all hate water, and that makes us want to be together. We all hate Tom Brady, so we're going to root for the same team together. Hydrophobic attractions. Some parts of your protein form and squeeze together because we all hate the same thing. We all hate water. Yeah, I like that, Laura. Brady-phobic. That's a good one. I might have to use that next semester when I offend somebody else about Tom Brady. So first way that the functional groups can impact the 3D shape of a protein, if they're hydrophobic or if they're nonpolar, they're going to squeeze together. Some parts of my protein keep its shape because all of us want to stay away from water. We'll make a little hydrophobic pocket, and that makes us happy. Some parts of my protein will form their 3D shape because those functional groups make hydrogen bonds with each other. Now, let's see if we can figure this out here. I've got some functional groups on my amino acids that are forming hydrogen bonds. Remember that we had 4 types of amino acid functional groups. We had polar, nonpolar, positive charge, and negative charge. These ones over here that are forming a hydrogen bond together, what would I classify these amino acids as? Which group would these amino acids be in, since they can make hydrogen bonds with each other? Yeah, several of us are chiming in. These would be examples of polar amino acids. Polar amino acids, the ones that have dipoles, partial positives and partial negatives, they can form hydrogen bonds with each other. So part of the reason I get this 3D shape for my protein is there's a polar amino acid over here that interacts with a polar amino acid over here. They make a hydrogen bond, and suddenly we're stuck together in this 3D shape. So polar amino acids work together to make these hydrogen bonds to stabilize my 3D shape. Non-polar amino acids squeeze together because of these hydrophobic attractions. Hey, let's talk about these ones down here. These two amino acids, I've got one with a positive charge and one with a negative charge. These two amino acids, because they have opposite charges, will actually form an ionic bond with each other. So some parts of my 3D structure that I see over here, they're being held together by the fact that I've got a negative in one part and a positive in another part, and they form an ionic bond together. Now check this out. One of my amino acids has a functional group with sulfur in it. With sulfur, I can see it here. Sulfur really likes to form chemical bonds with itself. Sulfur really likes to build what are called disulfide bonds, where two sulfurs interact with each other. A disulfide bond is an example of a non-polar covalent bond. Remind me, when we talked about non-polar covalent bonds, were those strong or weak? Non-polar covalent bonds. Yeah, those are really strong, right? Non-polar covalent bonds, those are really strong. So this right here, I'm gonna put a little 1 next to it. These disulfide bonds that I find between, it's an amino acid called cysteine. When this makes bonds with itself across parts of my amino acid chain, this is as strong as it gets. I'm never going to be able to break these bonds. Like, they're sharing electrons equally. These just don't break. When we talked about ionic bonds, did we say that those ones were strong or weak? What did we say about ionic bonds? Maybe in comparison to when we put water around them. Let's think about that. Yeah, ionic bonds don't do so great at surviving water. They're not a sharing bond. It's a stealing bond. So that's why somebody's positive, somebody's negative. So these are going to be considered a weaker bond where they may not hold on forever. Remember that hydrogen bonds, they were formed with dipoles, and dipoles are kind of constantly coming and going. So these ones aren't very strong either. These are just an attraction. Remember, we all hate Tom Brady, or we all hate water. So these aren't really a bond either. But all of these factors come together to take your alpha helices and your beta sheets to squeeze them into this shape and keep them in this shape. So all of these things, hydrogen bonds, hydrophobic attractions, these disulfide bonds or ionic bonds, all of these things come together to give your protein its tertiary structure, its 3D shape. And remind me again, let me draw my ugly amino acid again. Is it the amine group, the acid group, ooh, this is just terrible, or the functional group that gives us our 3D shape? Which part of my amino acid gives me my tertiary structure? Yeah, it's that functional group, right? So this part down here is responsible for my 3D structure, my tertiary structure. When I talk about making my primary structure, when I connect one amine group to another acid group, what was the special name of those chemical bonds that I formed between these parts of my amino acids to make primary structure? Yeah, it's those peptide bonds, right? Yep, so peptide bonds hold together the amines and the acids. And when we talk about secondary structure, remember that that was also those amine groups and those acid groups interacting with each other. It's just that it was the amine group here interacting with the acid group up here. When the amine groups and the acids interact at a distance, it makes that secondary structure. When they interact right next to each other, it makes the primary structure. It makes the line. And when those functional groups get going, we get that 3D shape of our proteins. You have a learning objective on the first page of your packet that talks about figuring out how we can use the chemistry of an amino acid to predict where it would be found in the plasma membrane. And that sounds super scary, but I promise it's not as scary as it sounds. Basically, what I'm asking you to do is to think about the chemistry words that we use to describe our parts of the membrane and apply that to which kinds of amino acids could live inside of it. So let's talk about our membrane. Remember that we've got these dots that are called the head groups, right? And we've got these tails that we have in the middle. When we talked about the heads and the tails, which of these parts, the heads or the tails, likes to be around water? Was it the heads or the tails who like water? Who likes water? Yeah, those heads like water, right? The heads like water. And remind me again, what's our big long word for I like water? Or maybe more technically, I love water. What's that chemistry word for I love water? Yeah, absolutely. Hydrophilic. Hydrophilic. Head groups are hydrophilic. They love water, which is a good thing because I got a bunch of water extracellularly outside the cell and a bunch of water inside the cytosol on the cytoplasmic side. So I've got these water-loving parts on the outside. My tails, what are their thoughts about water? How do the tails feel about water? Yeah, they don't like water. Yeah, I like that. Heck no, we're not doing the water thing today. No, thank you. Hydrophobic. Okay, so the middle of my membrane is hydrophobic. The outside of my membrane is hydrophilic. When we talked about amino acids, we didn't technically use the words hydrophobic and hydrophilic. We used the words polar and nonpolar. We use the words positively and negatively charged. When we're thinking about the membrane, the kinds of amino acids that can be embedded in the middle of the membrane with the tails, they're going to be the ones that are hydrophobic. What was the other word for hydrophobic that we use to describe amino acids? What else means hydrophobic? What's that other word? Yeah, exactly. Non-polar. Non-polar. Hydrophilic, my other word for that is polar, right? Polar. When you look at this picture, we've got some magenta or purple, whatever color we're going to call this. We've got some amino acids that are this purple magenta color. We've got some amino acids that are blue. The blue amino acids in this picture are right next to the tails in the plasma membrane. The blue amino acids, the ones that are embedded actually in the membrane, that are inside the membrane, This is going to be the ones that match the inside of the membrane, the ones that are non-polar. So non-polar amino acids live here in the middle of the membrane. Otherwise, if you live on the inside of the membrane or on the outside of the membrane, you're going to match the head groups because you're in the same place as them. If you live on the outside or the inside of that membrane, you are touching water. So you're going to either be polar, or you might be positive, or you might be negative. Here's where we tie it all together. Remember in unit number 1, where I told you, I promised you, that on the exam, you would have a question about what can and can't get across the membrane. And I was going to use words like this, right? Hydrophilic, polar, hydrophobic, nonpolar, all that jazz. I told you you don't care what the name of the molecule is. You just care about its chemistry word. You care about how it feels about water or how it feels about lipids. The same thing applies when I ask you about amino acids and I say, who could live inside the membrane? Or I say, who would live on the outside surface or the inside surface? You don't care about their names. You don't care if they're called serine or alanine or valine. You don't care. All you care about is, hey, this one is nonpolar, or this one is polar, or this one has a positive charge. All I need to know is their chemistry, and that tells me if they can live with those tails or if they're going to get kicked out, if they've got to live on the inside or the outside. So just like we use the chemistry of the plasma membrane to decide what could get in or out, could pass straight through it, you better believe if you're going to live inside the membrane with those tails, you better match those tails if you live there in the inside with those tails. I just talked a whole bunch. I need to pause for a moment. How do we feel about using these kinds of words to predict where amino acids would be found? Thumbs up, thumbs down. I guess I kind of did the bait and switch on you, didn't I? I said we were going to leave chemistry behind us, but actually can't ever leave chemistry behind us. I wish we could. Not my favorite, but we learn to live with it. All right, so we've talked about primary structure, the order that the amino acids go in. We've talked about secondary structure, when little parts of that amino acid chain fold on themselves to make curlicues or to make accordions. Then we talked about the 3D structure, where those functional groups tell us essentially where you can live in a protein. All of those things are structures that every single protein has. We're going to talk about now a level of structure that not every protein has. Some of them do, but not all of them do. So our last level of structure, not everybody has. This last level of structure is what we call quaternary structure. Quaternary structure. And this is the biggest, baddest level of structure that we can have in a protein. Yeah, the example that I'm going to use for you to show you quaternary structure is hemoglobin. But here, I want to make sure I clarify something for you all. When we talk about quaternary structure, hemoglobin is not the only protein that has quaternary structure. To define what quaternary structure is, all it means is more than one, I'm going to abbreviate, amino acid chain interacts. All that quaternary structure means is I've got more than one amino acid chain that was folded together. We put them all together to make like this superstructure. So remember, we keep talking about our super banana that's all souped up. Quaternary structure of proteins, they've got all the moving parts, all the working parts. So the example in your notes is hemoglobin, but I'll mention next lecture, so lesson number 7, we're going to talk about an enzyme called RNA polymerase. That's another protein that has quaternary structure, so more than one amino acid chain in it. I'm trying to think about some of the other ones. There's a very similar protein called DNA polymerase. There's one called helicase. Most of the proteins in your body do have quaternary structure. So please don't think that hemoglobin is the only protein with quaternary structure. Lots of your proteins have this. Quaternary structure, again, means that there's more than one amino acid chain that's interacting with one another. So when I look at hemoglobin, that's this protein that I see over here, hemoglobin is actually made of 4 different amino acid chains. I've got these 2 chains that I call alpha chains and 2 that are called beta chains. Now, these are different from alpha helices and beta sheets. It's just the name of the whole protein chain there. So 2 alpha proteins, 2 beta proteins that interact with each other to make this really big hemoglobin protein that we see here. Does anyone happen to know, when we talk about hemoglobin, what's its job? What's the job of hemoglobin? The function of that protein. Yeah, Tian's totally right. Hemoglobin, oxygen transport, absolutely. This is the way that we help our red blood cells to send blood throughout your body using hemoglobin. A teaser for us in lesson number 6, we're actually going to talk about a disease where there's something wrong with hemoglobin. It's called sickle cell anemia. So this is what hemoglobin is supposed to look like when it's doing its job correctly. So I've got 4 different amino acid chains. All of them are folded correctly, so all of them have tertiary structure. They interact with each other to form this really big protein. Now, I know this picture down here is really funky looking. That's why I want to explain it to you. Scientists took the hemoglobin protein and they figured out exactly which amino acids in each of those chains that make up hemoglobin, which exact amino acids are interacting with each other to hold these things together. So over here in this alpha chain, the amino acid number 141, it's forming a chemical bond with a couple of the amino acids over here in the other alpha chain. Or this amino acid right here, amino acid number 99 in this particular protein. That's interacting with this specific amino acid in the alpha protein over here. All of these different chemical bonds that these amino acids make with each other hold together my 4 amino acid chains. All 4 of these chains need to be the correct shape for them to be able to form these chemical bonds to hold it together. Because, hey, when we're talking about proteins and their shape, you all told me that's the anatomy of a protein. When I talk about the anatomy of a protein, how does that relate to its physiology? How does the anatomy of a protein relate to its physiology? Are they related? Does one impact the other, or are they totally separate? Yeah, they're definitely related to each other. Absolutely. Remember, we said in lesson number 1, anatomy dictates physiology, right? So I've got to have all 4 of these protein chains with the right anatomy with the right shape to be able to do the right job. And in the case of hemoglobin, that job is transporting oxygen. Let's talk about what it's like when I don't have the right shape. When a protein doesn't have the right shape anymore, we call it denatured. That's an underline highlight star word for us, denatured. A denatured protein is a protein that no longer has its correct shape. Hey, so not a trick question. If I don't have the correct shape, do you think I can do the correct job? If my shape is wrong, can I still do my job? Yeah, absolutely not, right? Anatomy dictates physiology. If I mess up my shape, I mess up my job too. When a protein is denatured, it has permanently lost its shape. Denatured proteins permanently lose their shape. There's no going back from there. A great way for you to visualize what denatured looks like for a protein is looking at eggs. So you heat up eggs and you go from having this clear part of the egg to what turns into a white part of the egg. We literally go from a protein you can't see to a protein that you can see because we changed the shape of the protein that's inside of it. The albumin protein, it becomes denatured. You can't ever take the whites of an egg and make them clear again. It just doesn't work. It's permanently changed. For those of us who had a chance to work on this lesson, do we remember what some of the things are that we can use to denature a protein? How do proteins get denatured? Do we remember? Yeah, so I gave you one of them, right, with our example with the eggs. Temperature is a big one. So if we make things really hot is the best way to do it, is an easy way to do it. Yeah, so if we make our pH wrong, so that means that either it is acidic or it's basic. If my pH is wrong, that could totally screw up my protein shape. Yeah, I can expose it to some chemicals. So if those chemicals break down peptide bonds or mess up those functional groups. Yeah, and Gerardo mentioned for us the last one, which is perhaps the scariest one, and that is other denatured proteins. If I have a protein that's already the wrong shape, sometimes it can go and bump into a neighbor protein and denature that protein as well. So if we make it too hot, if it's too acidic or basic, if I expose a protein to chemicals, all of those things can make it permanently lose its shape. In the body, one of the most important ways, though, that denatured proteins exist in us is if we bump into other denatured proteins. So let's talk about what this looks like. You have this picture, I think it's the last page in the lesson number 5 lesson, and it shows you two forms of a protein that we find inside neurons. Neurons, by the way, where do I find that kind of cell? Where do we find neurons in the body? Yeah, exactly. Neurons are the big cells that make up the brain. So we're looking at, over here on the left, we're looking at the normal protein that we find in these neurons. Over here on the right, we're looking at what we call the prion protein, prion version. So we're just going to call it a prion. This word prion is referring to a protein that has become infectious. And you're going to read a little bit more about this in your application activity for this lesson. But a prion is a protein that is infectious, or it has the ability to mess up normal proteins. So here's how this works. We start with a message. This is lesson number 6 here. We start with a message to build a protein that looks just like this. So I've got this nice, beautiful curly Q protein that I've made. Well, this protein's floating around in a neuron, and up comes this one down here. Up comes my prion, and my prion says, hey, you know, my friend, you would look really great if you had my shape. Can you all tell? I know the picture is a little weird. Can we tell which secondary structure this prion has a lot of? What secondary structure do we see here? Can we tell? We can back up to, there's only two options for secondary structure. Yeah, so a couple of us have identified it. The one that we see here was that one called the beta sheets. Beta sheets. So I'm seeing all of these flat spaces. These are the accordions. My prion proteins have a whole lot of accordion folds in them. Compared to my normal protein, see how over here my normal protein has all of these alpha helices? Lots of alpha helices is normal. My prion protein is the bad boy. The bad boy protein with all its beta sheets comes over to the regular protein and says, hey, you would look great with a beta sheet. And the regular protein says, sure, I kind of like that. Let's give it a try. And the regular protein changes its shape, or it's misfolded into a prion protein. The worst part, though, is now instead of just having one misfolded protein, now I've got two misfolded proteins. And both of these misfolded proteins are trolling around inside neurons, trying to find other ones that are the right shape. Eventually, so many of these misfolded proteins build up inside a neuron that the neuron ends up dying. So we're going to learn about, in our application activity, an example of this called Crewsfeldjacobs disease, where we take a normal protein like this, and we keep bumping it and bumping it and bumping it into these prion proteins until there's so many of these prion proteins inside your neurons that they can't function anymore. They can't do their job correctly anymore. So when we talk about prion proteins, these are proteins that are denatured that have the ability to denature other proteins. And remind me, if I am denatured, if I've permanently lost my shape, can I still do my job? If I've permanently lost my shape? Yeah, I've got to have my shape to do my job, right? Without that correct folding, without this pattern here, I can't do my job anymore. Yeah, so Fanchin was asking a little bit about the mechanism that we change things. Yeah, I think that some of it has to do with changing the bonds, changing the arrangement of those amino acids. So when this one comes over and kind of attaches to this one, it reorients some of that chemistry. So some of those hydrogen bonds, some of those ionic bonds, if we mess with enough of them, we can fold them into a new shape and make this prion. By the way, we said that this protein right here had a lot of beta sheets, right? How have any of you ever heard the terminology beta amyloid plaques? Has anyone ever heard of those before? Beta amyloid plaques. Okay, so here's where I'm going to be the mean teacher, and I'm going to make you do a little bit of digging. Beta amyloid plaques. Let me write it on the screen here. These are a form of protein. It's a protein that has a whole bunch of beta sheets. We see these form in a, common neurodegenerative disease. So, and I may have spelled it wrong. Now that I'm looking at it, I'm not quite sure. But there is a brain disease, this, aside from Crewsfeldjacobs, which we're going to study, a much more common brain disease where your brain cells fill up with these beta amyloid plaques, and you start to lose your ability to think and lose your ability to remember. Has anyone had mad Google skills and figured out what it was? Exactly. Yep. Beta amyloid plaques. That's when I build up a whole bunch of these amyloid proteins, or these beta amyloid proteins. We change everybody to be this non-functional beta sheet form, and it kills our neurons. That's the problem that we see in Alzheimer's. So another example of a misfolded protein at work, Alzheimer's disease. All right, that is all I've got for today. I think we will plan to just dive straight into lesson number 6 tomorrow. What you have left to do for lesson number 5, which is due on Sunday, lesson number 5 stuff due on Sunday. We have your learning activity that's specifically related to our lecture lesson. And then you also have the Crewsfeldjacobs learning activity to do as well. So like our bulletin board said, it's kind of the snarky way to talk about it is the human form of mad cow disease. But the way that it progresses in humans, the mechanism behind it is exactly the same as the way that it progresses in cows. They're in mad cow disease. So you need to go through and read. There are a couple of short videos for you to watch that tell you a little bit about it. There's a website with some questions, and then there's a learning activity. All that information about Creutzfeldt-Jacobs disease will be included on exam number 2. So don't forget about that assignment, and don't forget about completing and refreshing that before exam number 2. Any last minute questions today before we finish off our recording? Our minds are all blown, right? For multiple reasons. Our minds are blown. I will give us our daily penguin because I have been slacking on that lately. Our daily penguin is taking hold of the right shaped protein, normal protein. No prions. Yeah, Laura says no questions, but it's a lot to process. Absolutely. Yeah, so take your time to really think through it and process through it. I will get this video posted as soon as Blackboard is done with it. Other than that, I guess we will call it good. Feel free to reach out via email with any questions that you have about this content or any other stuff. Otherwise, I hope to see most of you back tomorrow when we start talking about lesson number 6.